Okay, can somebody confirm that you can hear me?
Yes, we can hear you.
Yes, we can hear you.
Great. Are there any questions before we begin? Excuse me here. All right. Let me state that, I thought so you should be seeing my screen now. I did a lot of grading, so take a look at what's been graded and what's not been graded. And I made an announcement. When did I go to Clark website? So take a look at that announcement. There's a new announcement. There's a bug in the Canvas website. And I think there's even a message when you first log into Canvas. But so far it has not hit us. But if you do notice a problem when you submit your assignment, it goes through steps to say what you can do. I think what you have to do is submit it again. There's the announcement, which I gave the same announcement. Our worksheet for the lecture is using a discussion board. So this may be a problem with us in the future, but at the moment, it was not a problem, meaning that when I went to the speed grader and the grade book, everybody's assignment did show up. Any question about any of that? I did want to point out another announcement. Let me get to the class website. Probably should open that another one. And that is Clark College has resources of the week. Every week there will be a new resource put up. I'm sure you're familiar with this, but I have a link to these resources. And so just click on this link and you'll come to the resources of the week. Well, this is confusing. I guess I haven't updated this. All right. Let me see if I can get to it from here. I'm not even sure. It's like they might not have updated it yet, which is odd. Where am I going? All right, I'll look into that and see. This looks like it's going to a previous term's resources of the week. And that's not what I intended that to do. College instructure. Yeah, that might be the wrong link. Anyways, I'll put a link here for the resources of the week, and then you can see the resources. And there probably will be more than one, sort of like this. This is the wrong term. This is for spring term, but there's different resources. All right. Oh, I don't want to go back to home. No. That's why I should have opened that in a different window. Oh, here, it was there, okay. Everybody did well on the plagiarism tutorial and plagiarism quiz, so that's good. Remember to use what you learned in the plagiarism quiz for your assignments, for the long assignments like the lab report and the infectious disease project. You'll be needing that information. All right, let me go to the syllabus. And I'm pretty sure we're working on chapters 1 and 10, but let me see if we're supposed to finish that today. Oh, that's rather loud. Yeah, it's really loud. All right, so yes, we are supposed to finish chapter 1. We'll see if we get finished with chapter 1 and start chapter 2. Any questions? We're waiting for this to load. No, I don't think that's where we were. I think I looked that up. Grading. Yeah, here we are. We were talking about the 3 domains, and then there are 5 kingdoms. And for some of the kingdoms, we're going to talk about the kingdom. Well, we already talked about the kingdom Protista, that you can split it into plant-like protists, the animal-like protists or the protozoa, and then the fungus-like protozoa. The fungus-like protists, sorry, I can get that right. Let's talk a little bit about the kingdom plants. We can separate the plants into different divisions. There are mosses, ferns, conifers, and the flowering plants. We're not going to talk much about the plants because they do not cause human disease, but all of the plants are multicellular. So all organisms which are a plant are multicellular. And that's all we're going to talk about the plants. Any questions? We'll talk a little bit more about the animals because there are some animals which do cause human disease. We can separate the animals into sponges, the worms, and the scientific name for that is helminth, and then the insects and the vertebrates. Once again, all animals are multicellular. For the animals which cause human disease, all of them are worms or helminths. Any question about any of that? Insects can plague people and they can give people disease, but like when a tick bites you and then you come down with some tick disease, it's not a disease from the tick per se, it's a disease from a bacteria or other microorganism that the tick carries, and then the tick when it bites you, you get the disease. Okay, so the helminths can cause disease. We're talking about a worm infestation. Insects can give us disease, but they themselves do not cause the disease. The exception would be something like somebody who's just covered in fleas and they're suffering from flea bites. And we don't have too many people like that, but that's a problem with dogs occasionally. But generally, insects give us a microorganism that then causes disease. Another example of that would be malaria, where the mosquito bites us, giving us the malaria parasite. What comes under the umbrella of microorganism? I sort of hinted at this when I was talking about the worms. Obviously, bacteria and archaea come underneath a microorganism. And then we mentioned that viruses and prions do also, but they are not organisms, meaning they don't have cells. You can argue about whether these are alive or not. We're not going to get into that argument, but we'll just call them a special microorganism because they're not an organism. And then there's certain of the eukarya which cause disease. The protozoa cause disease. Certain molds and yeast cause disease. And they are microorganisms because they're single celled. Some algae are microorganisms. If they're single celled algae or a colonial algae, which is so small you need a microscope to see them, then they are a microorganism. However, when we're talking about the kelp that washes up on the Oregon or Washington coast, that is not a microorganism. Okay, so it's only some algae are microorganisms. And then the helminths are considered a microorganism. Why do you think the helminths are considered a microorganism? Because most adult worms are not microscopic, like the beef tapeworm is, let me see if I can remember, something like 6 meters long. Definitely not microscopic. So why do we consider the worms, the helminths, microorganisms? Anyone have a guess? The helminths are multicellular animals. There are many of them that are parasitic, and they're all, if they're parasitic, they're either a flatworm or a roundworm. They're not strictly microorganisms, at least the adult is not. However, some of the life stages of the helminths are microscopic. And usually the stage of the worm, which is the stage that infects people, is microscopic, meaning most people get infected by a helminth from a fertilized egg, which is microscopic. Any question about any of that? So some of the life cycle stages of a helminth are microorganisms. And then more than that, the stage, which is usually the infective stage of the worm for infecting a person, is usually a microorganism, microscopic. All right, let's talk just a little bit more about viruses. They are acellular, meaning they do not have cells. They have a central molecule, which is their genome, and it consists of either DNA or RNA, but it is never both. This is their genome, it's the core molecule. This core molecule is surrounded by a protein coat, In some viruses, that protein coat may be enclosed by a lipid envelope, but not all viruses have a lipid envelope. And when do they replicate? Viruses only replicate inside their host cell. So they have to infect a host cell in order to replicate. Viruses are not part of the 3-domain system because they're not really an organism because they don't have cells. Any question about any of that? All right, when we compare the 3 domains, the bacteria, the archaea, and the eukaryotes, we can see that the bacteria and the archaea have the same cell type. They're prokaryotes, and the eukaryotes have the eukaryote cell type. When we look at their chromosome, both the bacteria and the archaea typically have one circular chromosome, and the eukaryotes typically have many chromosomes which are linear. So if the DNA or chromosome is circular, that means it's in a circle, and the starting point is the same point as the ending point. If we look at the X chromosome, it will have a starting point which differs from the ending point. So it's a linear chromosome. The chromosomes can differ in other ways, like the bacteria have no histone proteins on the chromosome. They have no introns. We'll talk more about that later. And the eukaryotes and the archaea do have histones and introns. You should know that eukaryotes have histones and introns, and then the bacteria do not have histones and do not have introns. You don't need to know that for the archaea. Okay. If we look at the ribosomes, they will be different. The bacteria and the archaea have ribosomes which are smaller than the eukaryote ribosomes. The bacteria and the archaea have 70 S ribosomes. The S is the sedimentation coefficient where the ribosome sediments in an ultracentrifuge gradient. And they sediment at a smaller sedimentation coefficient than the 80S ribosomes of eukaryotes, which are larger. And you don't really need to know about the sedimentation coefficient. I'm just explaining what the S is. It's what microbiologists and biochemists use to measure ribosomes. When we look at the cell wall, if the bacteria has a cell wall, it's mainly composed of a molecule called peptidoglycan. If we look at the archaea and the eukaryotes, these cells have no peptidoglycan. Even if they have a cell wall, they have no peptidoglycan. Anyways, that's just a comparison of the 3 domains. Any question about any of that? If not, let's move on and talk a little bit about the brief history of microbiology. The history of microbiology largely concerned the debate over whether life spontaneously generated or whether it did not. And it also covers the golden age of microbiology from years 1857 to 1914. You don't need to know this. This is just when microbiology first arose. Okay. And essentially in the golden age, anything anybody studied was something new. And so they called it the golden age. This is the history of microbiology in a nutshell, the golden age being right here. When we're talking about the history of microbiology, some of it goes back into ancient times. Now, when I give a date, you do not need to know any of these dates except for the first date. And the first date is when we look at the fossil record, we can find ancestors of bacteria in the fossil record, which gives us the first evidence of life on Earth, which goes back more than 3.5 billion years ago. Any questions about any of that? That's the only date you need to know. You might need to know when I give you some of the events, you don't need to know the dates, but you should know the order, meaning that before the Mayans made a fermented beverage, which was cacao, a chocolate, before they could do that in 600 AD, there had to be the first written records of microorganisms. And the first written records of microorganisms were Babylonian clay tablets where they were writing recipes for making beer. Now, the Babylonians and the Mayans, for that matter, did not know that microorganisms were either fermenting the beverage or fermenting the beer, but it was actually the microorganisms that did that. The point is, before the Mayans could do that, the Babylonians had to do that. And before that, we had to have the first evidence of life on Earth. So you might need to know the relative events. Any question about any of that? But you do not need to know the dates except for the first date. Here we're looking at an actual fossil record from 3.5 billion years ago. A bacteria, or I should say an ancestor of a bacteria. The next date, which you don't need to know the date, the next event occurred in 1590, where Janssen developed the first compound microscope. We had to have the microscope invented before microbiology could begin. In 1665, we had the first use of the microscope on living life forms, which Robert Hooke did. He took the microscope and looked at plant cells, and he saw cells under the microscope. He saw little boxes. See if I can draw that. Close that down. He saw little boxes, if I can get this working. And it reminded him of the rooms in a monastery where the monks had their private rooms, mainly for sleeping. And so the monks called these rooms a cell. So in the monastery, there would be a hallway out here, and then there would be a door here. But the rooms were shaped like this, and they called them cells. And this is where Robert Hooke got the name cells, because what he was looking at under the microscope reminded him of the rooms in the monastery that the monks called cells. Any question about any of that? This marked the beginning of the cell theory, where all living things were composed of cells, meaning after 1665, when people started looking at living things, they started seeing cells. And so somebody came up with the theory that all living things are composed of cells. This was, the cell theory was actually written up by Virchow many years later. The above is not specifically microbiology because plants are not microorganisms, and Robert Hooke was looking at cells. Although they are microscopic, they're not a microbe. Let me skip on to the first microbiology. The first microbiology observation was by this guy here, Antony van Leeuwenhoek, and I'm not Dutch, so I could be butchering his name. He was actually the first amateur microbiologist where he looked at things and saw the first microbes. He looked at scrapings from teeth, rainwater, peppercorn infusions, the river water from the River Thames, and he saw the first microbes. And this he did, writing his observations from, I think this is his lifespan, 1673 to 1723. This marked the beginning of microbiology. Any question about any of that? Now, microbiology largely concerned the debate about whether microorganisms spontaneously generated, meaning they arose as a vital force came into inanimate objects and created a living life form. That was spontaneous generation. The other hypothesis was the theory or hypothesis of biogenesis, that all living life forms arise from another life form, like a human baby comes about from two parents, the theory of biogenesis. Well, from 1600 to 1800, the theory of spontaneous generation competed with the theory of biogenesis. Over a century ago, beliefs included things like you threw a log into the fire and a newt would come out. People would say, aha, spontaneous generation. Log in the fire spontaneously gave rise to the newt. And then you could see flies emerging from manure. Another aha, flies are spontaneously generating. And then maggots appearing in a decaying corpse. Most were logical, but not scientific. Some were amusing, such as somebody wrote up about when you put sweaty underwear in a wheat in a jar, it would spontaneously give rise to mice. In reality, what happened was the mice climbed into the jar to get the wheat, and then because of the jar, the size of the jar, the mice got trapped in the jar. They did not spontaneously generate. There's the article, somebody writing about the mice. A brief sidetrack. When we're talking about microorganisms, you can know that they're growing in a dish by seeing the turbidity. So this has no turbidity. This dish has turbidity in it, and that's the turbidity is from the growth of microorganisms. The first evidence proving or disproving the theory of spontaneous generation came from Francesco Redi, which is somebody you should know because his evidence was actually the first documented controlled scientific experiment. Francesco Redi took 3 jars and he sealed them, putting inside of them meat, and then looking to see if maggots arose in the meat. And in the sealed jar, there were no maggots. And people said, you sealed them so the vital force could not get in. So that's why there were no maggots. And he said, okay, I'm going to cover the jar with a fine wire mesh net and put the meat inside, and he got no maggots. And then he left 3 jars that he left open, and the meat was inside of it, and maggots appeared. And so this experiment does not support spontaneous generation, meaning that the maggots did not spontaneously generate inside the meat. And so this is the first evidence that spontaneous generation did not occur. And after Francesco Redi's experiment, people said that large multicellular organisms could not spontaneously generate. However, they said it did not disprove that microorganisms could not spontaneously generate. Let me go back to the spontaneous generation, and then we'll come back to that one slide of Louis Pasteur, not Louis Pasteur, Antoni van Leeuwenhoek. And that is, in this time, Rudolf Virchow gave rise, well, actually the theory of biogenesis was in existence, but he actually published it in 1861, that all living life form came from another life form. And you don't really need to know anything except that he published the theory of biogenesis. What happened was Louis Pasteur came on and he gave the theory, well, he gave the first evidence that disproved spontaneous generation in microorganisms. And he confirmed, therefore, the theory of biogenesis. Louis Pasteur did a number of things. And one of the things he did was he showed that microorganisms are present in the air. So if you had a dish that is open and you put broth in it, microorganisms could fall out of the air and then contaminate the broth and then grow in the broth. He also showed that microorganisms could be destroyed by heat. And then he did his famous swan neck experiments, disproving the theory of spontaneous generation. What he did was he put broth in a glass container shaped like this, and then he heated the vessel right here, and he bent the container into a swan neck. And this container is open to the air, and he then heated the container to sterilize it, to kill the microbes which were in either the media or fell in from the air. And then it was open to the outside so the vital force could get in. But the microbes, when they fell from the air, they would be caught right here by gravity, and they would not go up because of gravity. And that kept the broth sterile. And he could show that if you took this broth and you brought it down here, and then you brought it back, then the microorganisms would grow in the broth, meaning he showed that microorganisms only could come about from pre-existing microorganisms. Any question about any of that? All right, let me go back to this right here. And that is Antoni van Leeuwenhoek built the best microscope of his days. And he actually literally grinded the lens himself. These microscopes were really kind of primitive by our standards. And that is, they're really only a super duper magnifying lens because he's only got one magnification lens. This we call a simple microscope. And he actually would then stick what he was observing on this pen right here and then hold it up to his eye like that to view the microorganisms. But his microscopes were so good that he was able to see cells that were so small that they were not seen again for another 100 years. Meaning here are his descriptions of cells he saw when he was looking at microorganisms, and you can probably recognize these. That looks like a rod, a bacteria. These are all rods. There's a bent rod, and those are cocci. There's a sporillum, meaning these are actually bacteria cells. Bacteria were not seen again after Antony van Leeuwenhoek's record for almost 100 years. What happened was in 100 years, the construction of the microscopes got better, and as they got better, their focusing became better, and then we were able to see bacteria under the compound microscope because the focusing just got better over 100 years. But in his day, nobody else could see bacteria. So he's the first person to ever view bacteria. And like I said, it took 100 years before somebody else could see bacteria using microscopes that were not his microscopes. Any question about any of that? All right. So with Antony van Leeuwenhoek and Louis Pasteur's work, people began to wonder about the relationship between microbes and disease. And some people theorized that microbes could cause disease. This became known as the germ theory of disease. Out of this work also came the work of immunity, meaning that we have our immune system, which helps protect us from germs or microbes. And then out of that also came the antimicrobial drug work. So we got antimicrobial drugs out of that. About this time, Louis Pasteur also developed the basic idea of aseptic technique, meaning that you could get a pure culture by using aseptic technique and then continue to grow it, that pure culture of containing only one species of bacteria, if you use aseptic technique. And this is something you will be learning in the lab. Hopefully you've already learned it. About that time, Koch developed the technique for streaking for isolation. This was important because it is an easy way to getting a pure culture. Louis Pasteur had a way of getting a pure culture, but it was really difficult to do. And Dr. Koch's way was much easier. You just streaked your bacteria on a solid media, and then you get isolated colonies. And you take the cells from one isolated colony and streak it out again. And if you get isolated colonies, you now put your cells through 2 colony isolation events. The chances are very good that you'll have a pure culture, greater than 95%. About this time, somebody working in Dr. Koch's lab, he was Dr. Koch's assistant, developed nutrient agar, a new medium for growing microbial microbes. And this was important because now we had a easy media to grow many microbes. And that was nutrient agar. And then about this time, Sergei Winogradsky developed the idea that microorganisms were involved in the biogeochemical cycles, such as the nitrogen cycle. The nitrogen moves around on the planet Earth because of microbes. And Sergei Winogradsky did further and showed that this happens also with the sulfur cycle and one other cycle, maybe the phosphorus cycle. I don't remember if it was phosphorus or carbon cycle, but it was one of those two. Sergei Winogradsky showed. Louis Pasteur at the same time showed that microbes were responsible for fermentation, such as alcoholic fermentation, where microorganisms take the sugar in juice, like grape juice, and can convert that sugar into alcohol of wine. And the microorganisms also do that in mead to make beer. Louis Pasteur went further and to show that it was actually other microorganisms that spoiled the beer and the wine. And what happened was another microorganism used the alcohol, so it digested the alcohol to make acetic acid, which then would spoil the beer and the wine. And what that alcohol was converted to is vinegar, acetic acid, spoiling the beer and the wine. Well, with Pasteur's work that microorganisms were spoiling beer and wine, and that further suggested that microorganisms may cause disease. And Louis Pasteur showed that the bacteria that spoils beer and wine could be removed by heating it at a temperature high enough to kill the spoilage microorganisms, but not hot enough to evaporate all of the alcohol in the wine. And he called this pasteurization, where you apply a high heat for short periods of time. And it's used in a wide variety of foods. Most of us know of it for pasteurization of milk. But actually most beer is pasteurized. Coors beer is one that is not, it's filtered, but most beer is pasteurized. All right, the fermentation studies that microorganisms could ferment sugar and convert the wine, and then the alcohol in the wine, and then other microbes could convert the alcohol into acetic acid, gave rise to the question, might microbes have similar relationships with plant and animals, and could they cause disease? This is known as the germ theory of disease. This was an important advance because originally people believed that disease was caused by poisonous vapors. So that people believed when somebody got cholera from walking through the swamp, it was because they were breathing in the poisonous vapors in the swamp. In reality, the swamp just happened to have the bacteria that cause cholera, and the people breathed it in from walking in the swamp. I'm not going to talk about that. You can read about it. We'll talk about the first person to show that a disease was caused by a microorganism was done by Robert Koch. And he proved that the bacterium Bacillus anthracis causes the disease anthrax. He then later showed the steps he used to prove that Bacillus anthracis causes the disease anthrax. And he published those steps and used them for proof that a given microbe causes a specific disease. And these we call Koch's postulates. This is a concept so important, we'll talk about it 3 different times in this term. And it's the steps you use to prove that a given germ causes a given disease. And he published that in 1884, Koch's postulates, the steps you use to prove that a given microorganism causes a given disease. Any question about any of that? Robert Koch was very lucky, and that is he happened to use a bacteria that was very large, and then it had structure to it, even under his primitive staining and primitive microscopes, meaning in his day when he was doing this research, the stains weren't developed. And so when people looked at microorganisms, they all looked the same. But he was able to use a very large bacteria that actually had some structure to it, meaning Bacillus anthracis could form endospores, which you can see something inside the bacteria. And that's why he was able to do this. And he's the first person to show that a given disease was caused by a given microorganism. And we'll talk more about that later in the term. Any questions about any of that? All right. A little earlier than this, but around the same time, we have the first vaccine, which was started by Edward Jenner. The first vaccination was against the disease smallpox. What Edward Jenner did was he got cowpox from a person and then inoculated another person with cowpox. And then he observed that the person inoculated with cowpox was protected against the disease smallpox. Now, in Edward Jenner's day, nobody understood why cowpox was protecting against the disease smallpox. And because of that, nobody was able to make any further vaccines. It wasn't until Louis Pasteur experimented with vaccinations or a vaccine that we understood what was going on. And that is cowpox was similar enough to smallpox that when the immune system responded to cowpox, some of that response would also act on smallpox, and that would give protection against the disease smallpox. And then after Louis Pasteur's work, other vaccines were made. One that you might know is Louis Pasteur made a vaccine against rabies, the disease rabies. The term vaccination is actually derived from the word vacca, which is a word meaning cow, from the first vaccine, which was using cowpox as the vaccine. And the protection the person gets from the vaccine is called immunity. Any question about any of that? All right. Chemotherapy is literally treatment of a disease with chemicals. However, today, the term chemotherapy is almost only used to mean using anti-cancer drugs. But the broader sense of the term chemotherapy and the original use of the word meant treatment of a disease with chemicals. We won't use that term in this class. We will use chemotherapeutic agent for treatment of a disease with a chemical. And a chemotherapeutic agent may be a synthetic drug, that's one that's man-made, or it could be an antibiotic. Now, strictly speaking, the textbook definition of an antibiotic is a chemical produced by a living microorganism that inhibits or kills other microorganisms. So by this textbook definition, penicillin G is an antibiotic because it's made by a living organism and it kills off other microbes, bacteria. However, let me see if I can remember this. Another member of the penicillin, carbacillin, is that the name? Whatever, another member of the penicillin family that is man-made, carboxylin, something like that, is a member of the penicillin family, so it's similar in structure to penicillin G, but it's man-made, so it is a synthetic drug, not an antibiotic by the textbook definition. Now, that is not the way we use the term antibiotic in popular common usage. And so I won't use the textbook definition for what is an antibiotic. Meaning you go out into the clinics and you'll even have doctors saying, I'm going to give you carboxylin, and they'll say it's an antibiotic. Okay. And this is simply meaning an antibacterial drug. The first chemotherapeutic agent that was ever available to Western Europe or modern Western medicine was actually quinine from the bark of the cinchona tree. And the bark contained quinine, and it was used to treat the drug malaria. The first drug that was found by people looking at for a drug happened in 1910, where Paul Ehrlich was just looking at different chemicals, and he had to look at about 500 chemicals before he found one that could treat the disease syphilis. And he found, quote, the first magic bullet, the first chemotherapeutic agent that could be used to treat an infectious disease. And that happened in 1910, where he found salvarsan, a synthetic arsenic drug that actually could treat the first known disease, or the first drug that was able to treat a disease, and that disease was syphilis. Any question about any of that? Well, there's a question for you. When was quinine, do you think, discovered and was first used to treat the disease malaria? When do you think that was? In the early 1900s, the late 1800s, the early 1800s, or some other time? When do you think it was?
Late 1800s?
The 1800s. Anybody else?
I was going to say early 1900s.
You're both wrong. It was actually in the mid 1600s, where Jesuit missionaries saw the natives of Peru eating the bark of the cinchona tree to treat their malaria. And then the missionaries noticed it worked. And so they sent the bark of the cinchona tree to Europe to treat malaria way back in the mid 1600s. And that was the first chemotherapeutic agent that Western medicine had. And then the next one didn't happen until 1910. The first antibiotic was discovered by Alexander Fleming in 1928. What he did was he streaked out his plate with bacteria, and he was sloppy, and he got contamination on the plate. And then he grew the plate at room temperature, where both the bacteria grew and the mold grew. And then he came back over the weekend and he looked at the plate, and what happened was he just forgot to put it in the incubator. And he noticed that around the mold colony, there was a region where the bacteria did not grow. And he correctly hypothesized that something was coming out of the mold, which was preventing the growth of the bacteria. And he called this an antibiotic, or I should say, we call it an antibiotic. Actually, I don't think he came up with the term. That term came up with somebody after him. He called it penicillin G, and that was the antibiotic coming out of the penicillium mold. It was not the first antibiotic that became clinically available because it was very difficult to grow up the mold and then isolate the, grow it up and then isolate the penicillin G from the media. That didn't happen until World War II, so in the 1940s. But the first antibiotic that became clinically available was sulfonamide, a sulfo drug. And it actually was a man-made drug. And so they were able to make it and then make it chemically pure. So it was very easy to make it clinically available. Penicillin was the next drug that became clinically available, and it was mass produced in the early 1940s. All right. When we're talking about microbes and human disease, you should realize that most microbes do not cause disease, that we have a large number of microbes living with us, and that we call the microbiota, which is a new term. If you ever hear me calling it the microflora, this is an old term, and it's because bacteria were once classified as plants, and hence the term flora for flora, flowers for plants that we use for microbes. I'll try not to use that, but when I took microbiology, we called it the normal microflora, and now we call it the normal microbiota, the microbes that are normally present in and on a person. Animals and plants are host to symbiotic microbial communities that are present, and they're actually central to the animal or plant's welfare. We have a large number of microbes living with us, and they're usually found on the skin, the oral and nasal cavities, the respiratory tract, the digestive tract, and the urethra. They're usually not found internally, such as internal organs in the blood, the heart, the kidneys, for example. They're not usually found there. We have an immune system, and if you get an infection, it will fight off the infection and remove it from the blood and the internal organs. The normal microbiota actually helps us. It can prevent the growth of the human pathogen from getting established on you by competing for resources that are needed by the human pathogen. The normal microbiota can secrete toxins that inhibit the growth of the pathogen. We'll talk a little bit about that later. The normal microbiota can also produce nutrients and growth factors, which help us, such as folic acid and vitamin K. We get a significant portion of folic acid and vitamin K from the microbes in our gut or intestine. And then microbes are central to our digestion, like E. coli helps us digest our food. Okay, this is particularly important for animals that eat plants like herbivores, where they have to have the microbes to digest the grass. We don't digest grass, but we still have microbes that help us digest our food. I'm not sure how important the microbes are in digesting our food, but let's just say that E. coli and other microbes help us digest our food. Any question about any of that? So most microbiota do not normally cause disease. They can cause disease under certain circumstances, such as you're immunocompromised. You can get your normal microbiota growing where it shouldn't be and then causing disease. Infectious disease is where a pathogen overcomes the host resistance and then grows in the host, causing disease. And this we call a pathogenic microbe, a microorganism capable of causing disease. And it'll cause an infectious disease, meaning if it's a microbe, it can spread from one organism to another, and that we call an infectious disease. Any question about any of that? All right. There are emerging infectious diseases, and these are new or changing diseases that are increasing or have the potential to increase in incidence. Can anyone state an emerging infectious disease right now?
Monkeypox?
Say again?
Monkeypox?
Monkeypox would be an emerging infectious disease. Another one would be COVID-19. It's still relatively new. I suppose it isn't increasing at the moment, but it's fairly new. So it is an emerging infectious disease. One that may emerge in the future would be something like bird flu. They're worried that it may hit humans and then spread among humans. At the moment, when a person becomes infected with bird flu, it doesn't spread person to person, but it could sometime in the future. And so it's considered an emerging infectious disease or potentially an emerging infectious disease. All right, that's all you need to know about an emerging infectious disease, and that is the term. I've got slides on emerging infectious disease, but I'm not going to ask any questions on them other than the term itself. Any question about any of that? So it's a new or changing disease that is either increasing in the recent past or is showing potential to increase in the near future. Emerging infectious diseases are caused by microbes, and it can be any microbe. All right, any question about any of that? If not, that's it for lesson 1. Let's briefly move on to lesson 2. we'll only get started because there's only about 12 more minutes of class. This slide, as always, shows you the major goals and the rough outline. Now remember, if you are looking for all of the goals, you should look at the objectives, which are given either in the start of the study guide, right there, or I'll break it off for the objectives and overview. Any question about any of that? You do need to know all of the objectives for taking the quizzes, but obviously I can't show all of the objectives on one slide. So I'm just showing you the major goals for the lesson. And then a rough outline, what we'll be talking about. We'll be talking about the different terms of this lesson. And then with respect to the 4 biological molecules, you should be able to describe the relationships and the characteristics of the monomers and the polymers, be able to describe the composition and structure and function of the 4 biological molecules, and then be able to give examples of each. Can anyone tell me what the 4 major biological molecules are? What's one of
I
them?
can.
Say again?
Carbohydrate. Sorry.
Carbohydrates is correct. What's the
Protein.
lipid, you'll see as fats on your cereal box. Protein, which you'll see on your cereal box. And then the 4th biological molecule you'll never see on your cereal box or in any label, and that would be nucleic acids. These molecules are only found in something that was alive or was once alive, why it's called a biological molecule. Meaning you'll never find the 4 biological molecules from a piece of moon rock. All right, in lesson 2, it's important to realize the scale of things. You do not need to know the size, but you should know the ranking, meaning a human is bigger than a chicken egg, which is bigger than a frog egg, which is bigger than a eukaryotic cell, which is bigger than bacteria cell. Oops, didn't mean to do that. But to do this, which is bigger, too far. Which is bigger than a virus. Prokaryotes bigger than a virus, which are bigger than a large molecule like a protein, which is bigger than a small molecule like water, which is bigger than an atom. So you should know the relative sizes of things. You should know that things from about a bacteria to a frog egg, you need a light microscope to see. Things smaller than that, you need an electron microscope to see. And then things bigger than that, the human eye can see. Any question about any of that? Oops, why didn't that work? There is one mistake in this slide, and I'm surprised the author never corrected this. And that is it has 1 micrometer twice, right here and right there. This one shouldn't be a micrometer, that should be a millimeter. I'll point out that in our next lesson. So when we're talking about chemistry and the biological molecules, you should know that the biological molecules are an organic compound. An organic compound is typically complex. So a structure is complex, and it's typically bound together with carbon, and carbon combined to 4 other atoms. And then the organic compounds are typically held together mostly by covalent chemical bonds. That differs from an inorganic compound, which typically lacks carbon. Inorganic compounds are typically simpler than an organic compound. And they may be required by living organisms to maintain their growth, maintain their repair, and maintain their body as well as reproduction. Some examples of an inorganic compound are carbon dioxide, oxygen, and water. Water is one of the most important and abundant molecules you can find in a living organism. So we'll talk a little bit about that in our next slide. And you should note that one of the examples I gave you of an inorganic compound, carbon dioxide, does contain carbon, but it is not an organic compound, mainly because it's simple. Carbon dioxide is a simple molecule, and an organic compound is much more complex. We will take a look at complexity, probably not today, but in our next lesson. Any question about any of this? All right. Water is extremely important to living organisms. Most living organisms are at least 70% water. You should know that water is a polar molecule, meaning it is partially charged. The hydrogen has less of the electrons than the oxygen, meaning the electron is pulled towards oxygen, so the electron is not shared equally between the oxygen and the hydrogen. That makes the oxygen partially negatively charged and the hydrogen partially positively charged. This is not a complete charge. A complete charge would be a -1 and a +1, and this is less than that. So it's a partial charge. And your textbook doesn't use the term partial charge. I forget what your textbook calls it. Anyways, you get the idea. The polarity of water is important because water can form a hydrogen bond, which is where the positive hydrogen of this water molecule is attracted to the negative oxygen of this water molecule. And this creates a hydrogen bond between the two water molecules, where this hydrogen is attracted to that negative, partially negatively charged oxygen. This means that water has 2 chemical bonds, a hydrogen bond between water molecules when we're talking about liquid water, and then within the water molecule, we have a covalent water bond, which is actually a polar covalent bond within the water molecule. Any question on any of that?
I wanted to let you know, Dr. Volt, that it is 6:18. Just wanted to let you know.
Okay.
That's all.
Yeah, we're going to finish after this slide. All right. There's a little video you can watch on the water molecule, and water is a polar molecule. We'll talk more about that in our next lesson. Any questions on anything? If not, I'm going to stop here and we will continue with this next time. Okay? All right, I'll see you in the lab at 6:30.